All right, let's dive right into this explainer. Today, we're going on a really fascinating journey, taking us all the way from wide-open macro ecosystems right down into the invisible, microscopic machinery of life. We're unpacking the dynamic world of Class 12 biology practicals, going step by step through the core experiments that actually define our modern understanding of the natural world. So the core syllabus here is made up of exactly 13 practical experiments. But it's important to realize these aren't just mere checklist items to get through. They're real gateways. Each of these 13 labs acts like a unique lens, letting us see exactly how the fundamental rules of biology govern literally everything, from the largest forests down to the smallest individual cells.
Just look at the sheer diversity of topics we cover here. It's incredible. We're spanning the entire biological spectrum, from isolating actual plant DNA and drafting pedigree charts to track genetic traits, all the way over to identifying disease-causing organisms like Ascaris and Plasmodium. We even get into mammalian biology, looking at blastula development, heart sections, the whole shebang. But for today, we're going to focus tightly on three main areas: tracking population ecology, observing pollen reproduction, and witnessing cellular mitosis.
So here's our roadmap for this explainer. We're starting out in the field to measure plant population density and frequency, and then we're heading indoors, zooming all the way in to the cellular level to check out pollen germination and onion root mitosis. Okay, part 1, measuring plant population density. Let's start with that macro view and see how we quantify these dynamic, constantly changing living populations.
Now, what's really fascinating here is how we distinguish this first ecological metric. Population density is basically a precise head count of a specific area at a specific time. Think about it. Populations are constantly fluctuating, right? Births, deaths, migration. So ecologists need a reliable way to get a snapshot of a habitat's current state. We do this by taking the total number of individuals we find and simply dividing it by the total number of sampling units we've studied. To actually pull this off out in the wild, ecologists use a genuinely brilliant tool called the quadrat method. Because, let's face it, trying to count every single blade of grass in a massive field, that's literally impossible. So instead, ecologists use a quadrat. It's usually a 1 square meter boundary marked out with nails and thread. Setting up this standardized subunit makes counting large habitats not only feasible, but scientifically rigorous.
Let's look at some actual field numbers. This observation matrix gives us undeniable proof of how density differs across species in a test area. Take species A, for example. Out of 10 quadrats we threw down, we found 21 individuals. That gives us a density of 2.1 plants per quadrat. Compare that to species D, where we only found 4 individuals in total, giving us a density of just 0.4. This mathematical breakdown tells us exactly who is thriving and who is just scraping by in this space.
Moving right along to our second metric, calculating plant population frequency. Let's see how this builds our understanding of species distribution beyond just a simple head count.
Here's the deal with frequency. It actually doesn't care about the total number of plants. You could have 100 plants tightly clumped together in one tiny corner of a field. Their density there would be super high, but their frequency? Incredibly low. Frequency measures how widely scattered and common a species is across the whole habitat. We figure this out just by noting whether it's present or absent in random, widely distributed sample areas.
Notice how species A appeared in every single one of the 10 quadrats we sampled? That gives it a 100% frequency. It is literally everywhere. But look at species C. It was only present in half the quadrats, so it gets a 50% frequency. This paints a totally different ecological picture than raw density does. It shows us how successfully a species has managed to geographically colonize its environment.
All right, time to shift gears. We're leaving the vast fields behind, heading indoors, and zooming way in under the compound microscope, observing pollen germination.
This procedural setup is basically a recipe to trick nature. We are tricking the pollen into germinating in a synthetic medium rather than on the sticky stigma of a plant where it naturally belongs. By meticulously mixing sucrose, potassium nitrate, magnesium sulfate, and boric acid, we create this perfect chemical illusion. We put a sweet, nutrient-rich droplet of it on a slide, dust fresh pollen right on top, and then we just wait for the magic to happen.
And when it works, you get to witness this incredible physical transformation. What happens is the inner wall of the pollen grain, which is called the intine, literally bursts its way right through the hard outer shell, the exene. It punches through to form a long, emerging pollen tube. Out in nature, this tube would tunnel all the way down the style of the flower just to reach the ovary. Keep in mind, these structures are unbelievably tiny, just 25 to 50 micrometers in diameter. But the absolutely crucial takeaway here is what's traveling inside that new tube. The generative cell from the pollen grain travels down that newly formed pathway and divides into two male gametes. It's this microscopic journey that is 100% necessary for plant life to propagate and for new seeds to form.
For our final stop, we are zooming in even further to witness the very engine of biological growth itself, studying onion root mitosis.
Quick refresher on the fundamental rules of cell division here. Somatic cells, like the ones making up your skin or an onion's root, use mitosis. This process pumps out identical replica cells that maintain the exact same number of chromosomes. It's perfect for everyday growth, maintenance, and repair. This is totally different from meiosis, which is a specialized division that actually halves the chromosomes specifically to create gametes for reproduction.
Now, to actually catch mitosis in action, we need tissue that's growing fast. Freshly sprouted onion roots are perfect for this, but the chemical steps are vital. We use an aceto-alcohol fixative and a really vivid acetocarmine stain. Why? Because we are effectively freezing those rapidly dividing cells exactly where they are in their life cycle. The stain makes their chromosomes pop out so they're highly visible against the rest of the cellular background.
Then we get to a really fun, kind of low-tech physical step. We literally squash the tissue. Actually, scratch that. We gently tap it. But the reason we do this is crucial. If we didn't, we'd just be staring at a dense, dark brick of overlapping plant matter. By squashing it, we force that meristematic tissue to spread out into a single, beautifully thin layer where we can analyze individual cells.
And once you get that under the microscope, it is amazing. You can literally watch the cell cycle unfold across its dynamic phases. It starts with interphase, where the parent cell is gathering nutrients and copying its DNA. Then prophase hits, and the chromosomes condense. In metaphase, they line up perfectly right down the middle. Anaphase dramatically pulls those replicated chromosomes apart to opposite sides. And finally, telophase pinches everything off to form two brand new functioning diploid daughter cells. Like I said, the engine of all biological growth.
So, let's bring this all together. From throwing down quadrats to mathematically track expansive plant populations out in an open field, all the way down to witnessing the microscopic division of a single onion cell. How did these deeply interconnected processes shape the living world around you? Grasping biology at these vastly different scales truly changes how we see the natural world, doesn't it? Thank you so much for joining me on this explainer. Keep your curiosity alive, and we'll catch you next time.